Thanks everyone for joining us. This is our quarterly lecture series from the Network for Ocean Worlds, the Research Coordination Network. This is season 4 that's been focused on making measurements on ocean worlds, and this will have a life detection flavor to today's pairing of both Earth and planetary science speakers. Today's speakers are Shannon MacKenzie from Johns Hopkins University Applied Physics Lab and Shujie Wang from the Pennsylvania State University. And they have a very nice pairing of talks of looking at both the Earth and hopefully Enceladus for signs of life.
So our first speaker is Dr. Shannon MacKenzie, as Erin mentioned, from the Johns Hopkins University Applied Physics Lab. She's a planetary scientist interested in the icy satellites of the outer solar system and the astrobiological opportunity they represent. She serves as the deputy project scientist on NASA's Dragonfly mission to Titan and served as the PI to the predecadal mission concept study into flagship level astrobiology missions at Enceladus. And today she'll be talking about the Enceladus Orbilander, a flagship mission concept to search for signs of life. So without further ado, Dr. Shannon MacKenzie.
All right, thank you very much for having me. I'm gonna apologize. I'm recovering from that dreaded virus we've been all avoiding. So apologies for any throat clearing as we go on here today. But I was very honored to be speaking about the Enceladus Orbilander. In case you don't know what that is, it is a mission concept that was put together in preparation for the decadal. So very important to get up front that Orbilander is a mission concept study, which means that it's our goal in putting this kind of mission concept together was really to show what's possible and advocate for science at Enceladus, not necessarily to advocate for any specific kind of instrument or very specific operation schedule. That's not the goal here. The goal here is to demonstrate what's possible in the next decade. So the kind of bottom line up front is that the Orbilander mission concept was recommended as the second highest priority flagship in this in the decadal survey that we're currently living in by the National Academies of Science. What's important to remember is that it's very exciting that Enceladus Orbilander is on this list, but of course above it is listed Uranus orbiter and probe, and before that is Mars sample return. So it's very exciting, but there's a lot of work to go before something like Orbilander will see fruition. So hopefully today in the talk we can start to dive into some of the ways that we might go back at Enceladus, but we've got a lot of time to refine those ideas and honestly probably improve them. So this all came about feeding into this round of the decadal survey, the 2023 to 2032. NASA put out a call for planetary mission concept studies to be done by the community before the decadal survey actually kicked off. So normally the decadal survey solicits these studies to happen as part of their debate process, and this Rose's call allowed the community and NASA to kind of have input into that before the committee was even selected, I think, at that point. So our team was specifically chosen to look at the question of why and how we might go back to Enceladus. And what we argued in our proposal is that there's really one major question, one driving question at Enceladus that we wanted to focus on, and that is Enceladus inhabited? Thanks to all of the phenomenal data that we have from Cassini, we are well poised to not, we should also be asking questions at Enceladus about whether Enceladus is habitable. But we are also able to go to that next step and ask the question of, is Enceladus inhabited or has it been inhabited? Really do a search for life mission at Enceladus. We have the scientific basis to make that leap, which is really exciting. We also wanted to, so that was our scientific focus. Our kind of mission scope that we wanted to investigate was the far side of this chart, the Flagship class mission. I have some numbers put on here that are kind of like the colloquial values that are thrown about for what is a New Frontiers or Discovery or Flagship, but the reality of those numbers is quite different, but this is how we kind of all agree to think about them. We specifically targeted Flagship because in previous New Frontiers, there have been studies put forward on how to go back to Enceladus and do kind of smaller scoped science on that scope, on that budget scope. And we wanted to say, okay, if we went into a Flagship specifically with the astrobiology questions as our driving focus, what would that look like? So in that tactic, we broke up the phase space into these four kind of options. What if you had an orbiter with your life detection suite on the orbiter? What if you had a lander and just went straight down to the surface of Enceladus and ran your life detection suite there? What if you had two elements, that's the bottom row here, and you had a large orbiter and a small lander? Or what if you invert that and have more like a relay station and send all your fancy equipment to the surface? So that was the scope of our study. And in deciding between those different architectures, we really had to focus on the science. And the goals here were really focused on the searching for signs of life. We targeted a combination of both chemical biosignatures that are well supported by the Cassini data, but perhaps especially in isolation, less convincing of, hey, there's life here. So that's why we also put on our list something that we called confirmation biosignatures. We're not crazy about the term, but it was a succinct way to convey what we mean, that these are higher risk, but also perhaps higher reward measurements that you could make to really solidify the argument of, yes, these data together are telling us that there is life at Enceladus. These science objectives mapped to a suite of instruments that we call the Life Detection Suite. And you'll notice that on the kind of going from the left to the right, we included things that are kind of the bread and butter of planetary exploration. Mass spectrometers have been thrown across the solar system to great effect. And then moving as you go to the right to things that are more on the forefront of development. I think that this payload is a good representation of what's possible, but it's certainly not an exhaustive representation. There's lots of really great instrumentation that you could consider. For example, separation capable mass spectrometers. GCMS is not the only option for doing that. So we were mostly picking things that picking instruments that we could put on our mission concept spacecraft to demonstrate what's possible, and are highly advocating that a plethora of different kinds of instruments are developed so that we can actually do a convincing life detection biosignature search. We think that this kind of suite of approaches is really important, that you have complementary and orthogonal measurements. Again, that's what we tried to build our payload. That's the philosophy we used to build our payload. But since we were targeting this Flagship class mission, we wanted to do Flagship class science and broaden our scope to include those really key remote sensing and in situ measurements that help set the shape of our understanding of the Enceladus Enceladus ocean as an environment, and therefore feeds into some of that interpretation of those biosignature search measurements. So this is things like determining the ejection mechanics, how might the plume particles be changing as they go from ocean to space, what that plumbing structure looked like, and also assessing the ocean habitability. Really, what are those driving environmental conditions, and how do they provide context for how we interpret those potential biosignature measurements? These science investigations also lend themselves to a suite of potential instruments. So again, these are the ones that we've listed here. There are probably other ways to do it, and whether or not they're better is something we could all sit around and debate until the cows come home, which is part of the fun. But this is to summarize the payload that we put together. And again, we were looking at different architectures, ones that were in orbit, ones that were on the surface. And what we settled on was this concept of the Orbilander. Orbilander being one word, where the same spacecraft that orbits then goes down to the surface and acts as a lander. The reason that this is possible is that Enceladus is so small relative to the body that it's orbiting around. Enceladus is only 500 kilometers in diameter, and I think you would need like 1,000 of them to span the diameter of Saturn. So because getting into Enceladus orbit is where you have to is the hardest part, where you have to shed the most delta V. Once you're there in orbit around Enceladus, it doesn't take quite as much to get down to the surface. That's where this Orbilander concept really came into being, was arising from the physics of the situation. So I've jumped ahead here accidentally. Once Orbilander would get in to orbit around Saturn, the concept of operations is that we would spend most of our time in this pedal orbit, where we are sending information back to Earth, sending all of our measurement data, but also like spacecraft telemetry back to Earth up in this upper part of the orbit. And then as we're approaching Enceladus again, doing station keeping maneuvers. Enceladus is so tiny that the constant tugging from Saturn means that you have to actively maintain that orbit. And then once we're getting really close to Enceladus with periapses between 20 and 70 kilometers, that's when we would do our primary science, all of our remote sensing and tasting the plume by collecting plume particles with a funnel or a plate and ingesting those for later analysis with our life detection suite. So that orbital phase, we estimated taking about 1.5 years, and then doing our landed phase in about 2 years. But the beauty of the CONOPS is that there is some wiggle room. Those tilde are real for a reason. If it takes us longer to identify a landing site that we really like in the remote sensing suite data, we have that time to do that and really make sure that it's the right place that we want to land, and then do our same life detection measurements, but from the surface. So why bother to get down to the surface when Enceladus has this plume of ice materials that's erupting into space and making the ocean available to us from space? Well, that's because the plume is not the same as you go from near the surface to out into space. The physics of ejection is kind of naturally segregating particles based on their size. So at the higher altitudes, you get a lot of vapor in the plume, and also micron to nanometer size ice grains. This is some of what Cassini was able to sample. But as you get lower, you're getting much larger ice grains, and we think that those largest ice grains are going to be the ones that have the highest organic content. So we want to be able to sample all of those and kind of build a transect of our understanding of the plume and therefore Enceladus's ocean. So we proposed collecting material 3 different ways: flying through and doing passive collection with a funnel, or a plate. And then once on the surface, you can use that same funnel or plate to catch material. And that means the material is less likely to be modified. But to get a large volume of material all at once, you can also just actively interrogate the surface with like a scoop or a rasp or something like that. So all of these three different sampling regimes kind of offer a different insight into the plume materials and therefore into the ocean. And it gets you at different volumes of material as well, which is important for thinking about how we can run these different instruments. Some of them require, like the nanopore, for example, requires a lot more material. If we're assuming low amounts of organics in the plume material, which you kind of have to be robust against. So taking into account these three sample collection mechanisms and our 5 biosignature investigations, we thought that something like the Orbilander would be able to detect life at levels up to 500,000 times scarcer than in Earth's oceans, which seemed like a pretty robust way to go back to Enceladus and try to get at that question of, is Enceladus inhabited? Yeah, we can just leave it here. Yeah.
Okay, great. And so I'll just encourage people to open the chat and throw your questions in there. And if you haven't had a chance to do that yet, while people are going, I'll sort of take my prerogative and ask a question, because I think as you sort of set the stage, right, there's a lot of community excitement around this mission, but we probably have some time before it really gets off the ground. So do you have sort of thoughts on what maybe the top either technology or kind of science developments that the community can focus on to, provide better constraints or, really help focus and make the mission more successful when it does happen, given that we have time for lab experiments or data mining or, something of that nature?
Oh, that's a great question, and always a tricky one. Pick your favorite child, right? Like, there are lots of different. One of the really revelatory things for me from this process was just how many phase spaces you could think about optimizing in. I think that we have a lot of work to go in this concept of a life detection suite. One of the things that we really relied on in our spacecraft design was simplifying the sample handling and processing, for example. And at the time, the way that these mission concept studies kind of work, you have to rely on instruments that have already been proposed or created as proof of concept. And what we we're really hoping to see is something, and I think this is more like what the Europa lander was envisioning, where you have a centralized system that's able to deliver sample downstream to multiple instruments. And if those instruments could look at the sample in series or in parallel as the different measurements might require, those are really powerful ways to do the science, but require a lot of coordination at like the mission level early on in the mission architecture definition. So we all need to be getting on board together as soon as possible. I don't entirely know how to make that happen, but if we keep talking about it, maybe it will.
Preaching to the choir. I appreciate it. Okay, so a question from the chat. At what levels would you call an ocean uninhabited? Could there still be a biosphere if there is less than the 500,000 times, lower than biomass on Earth?
Yeah, great question, Catherine. And a hint at how tricky our language and careful our language needs to be. I would say that if we didn't see anything, and we have one level of confidence in our detection, really all that means is that, yeah, there's there's not biomass here at this 5,000 times less value. And we can't say, like, how do you prove a negative, right? That's really hard. But I think even being able to say that if there's any biomass, it has to be this level of concentration would be really a powerful measurement, especially if tied together with other measurements of the environment. If we're able to kind of provide the context of why would biomass be so low / non-existent, like maybe we're not seeing the right products of the kinds of metabolisms we would be expecting, right? That would help kind of fill in our reasoning behind that result.
Thanks. So a more sort of ConOps and mission related question: how close to a plume source do you have to be for the funnel to collect a significant amount of material when you're in that landed phase? I guess when you're doing that passive, fall collection.
Yeah, that's a great question. Selecting a landing site is both a function of wanting to maximize how much material you can collect, but also maximize the probability that your spacecraft will survive landing. We don't want to land right next to the tiger stripes, for example, in case it's like the plume material has fallen. Yeah, there's a lot of plume material falling right next to the tiger stripes based on some of the really awesome modeling work that's been done. I think that paper came out in 2017. But really close to the vent, there are lots of boulders, and it's not clear. So in some areas there are lots of boulders, and in other areas it looks very smooth. But what if it's not compacted? What if it's just a thick layer of snow and your spacecraft is going to go whoop? That would be hard to send data back if you can't see the horizon because you've sunken into a pile of snow. So it's a tricky balance of maximizing how much plume material you can collect and minimizing the risk to the spacecraft. And that's why having that remote sensing suite, really interrogating the surface to a much higher fidelity than what we can do everywhere with Cassini now, is such an important part of this mission concept.
Thank you. So a slightly different question. Can geophysical instrumentation help understand the water flow or ice water interface on Enceladus? It's predicted that the ice thickness is less in the South Polar region.
Yes, it is my understanding as well that the crust is thinner at the poles. That was one of the things that we were targeting with a radar and sounding radar investigation, but that's not the only way. That's not the only geophysical instrumentation you could envision putting on a spacecraft to do important interrogations of what's going on there, especially at the South Pole, and trying to understand like the fracturing mechanics. I would love to see something like that on an Orbilander.
Great, thanks. And so there are more questions coming in, but I want to give us a chance to switch over to Shujie's talk. I guess, Shannon, you should feel free to try to answer some of those directly, or again, if we might have some time at the end where we can circle back and take a few more questions. So
cool. Thanks so much.
Yeah, with that, Audrey, if you want to just put up the introductory slides again, and Michelle will take it away. Yes, thank you. Applause, right. It's always forget to do that in virtual seminars, but yes, thank you so much, Shannon.
Okay, and our next speaker is Dr. Shujie Wang from the Pennsylvania State University Geography Department, who's going to discuss the use of ocean color satellites to detect glacier ice from space and characterize how their growth darkens the Greenland ice sheet surface. Dr. Wang is a cryospheric scientist focused on the understanding the processes controlling the mass balance of Earth's ice sheets, the factors governing the ice sheet stability, and the changes in the ice sheets over space and time in a warming world. So without further ado, Dr. Wang.
Thank you for inviting me here. I'm totally different background from this group, and I'm going to talk about mapping glacier algae use ocean color satellites over the Greenland ice sheet. I'm a geographer, so I'm very interested in mapping different kind of processes. So most of this work I'm going to present it was conducted when I was postdoc at Columbia University. And although I have actually switched my research direction quite a bit recently, but it's very nice to have this opportunity to present this work again. So it's interesting that to hear the new mission, flag mission, and it's also interesting for us as Earth scientists to know, how we can actually help build kind of analogs or Earth actually to study those planetary science. So I'm gonna switch our lens back to the Greenland ice sheet in Northern Hemisphere. So Greenland ice sheet is of great importance to ice and the climate community because we care about the mass loss of the Greenland ice sheet and also its contribution to the sea level rise. So most one aspect of the Greenland ice sheet, the mass loss is more than half of the mass loss is due to surface mass balance. So basically caused by surface melt and runoff. So one critical parameter to dominate this process is called albedo. So describe how much the solar radiation can be reflected by the surface. So this is particularly interest in southwest Greenland ice sheet. We can see a very dark ice band forms in every summer season. So we use satellite data, we can actually tracking the time sequence, how the bare ice and dark ice change in our summertime, in our summer season. So this kind of shows a map in the summer of 2017. Basically, the blue area means bare ice, means the snow cover melted out and exposed the ice layers. And then this bare ice expanded in the July after the melting peak happens in this area, and a dark ice zone would rapidly develop in this area. So it peaks in like late July to early August when the snowfall comes. Then this dark ice band just buried under snow. So one critical question is that what are those dark materials? And of course, like we study the earth science, we kind of cannot really complain we don't have enough in situ data, but we also have very limited observations in this area. Until recently, more and more field studies find out those dark materials could be glacier algae blooms. So this is some very cool field observations conducted by Black Bloom and Deep Purple projects in Europe. They collected some amazing field data sets on the surface of those dark materials. So this is a drone image they took over the field site. If you zoom in the surface, you can see those very brown dark material scattered on the surface material. So if you look at the under microscope, they can find different algae species actually generate those brown color pigments and darkening the surface. So those algae, because they generate those dark brown pigments, they can reduce albedo. So those field measurements basically shows that those if the population of the algae doubles, then they can reduce albedo like 4%. Actually, for the glacier algae documentation, it's not very recent. It actually can date back to 1872, and during an expedition, they found this kind of material, and they kind of write it as a brown polycellular algae on the ice surface. So as a geographer, I'm very interested in can we actually mapping the distribution of this glacier algae use satellites data? Because even though we can get very cool field observations, but we still have very limited observations across the space and time to study the evolution of those algae blooms in a melting season, and also how if there's any annual variability of these algae blooms. So to map the glacier algae, first thing we need to know what's their spectral signature. And we have so many sensors orbit, we have to know what kind of sensors can respond to those spectral signatures. So you may also heard about snow algae. So snow algae, quite different from glacier algae. Snow algae, what we also called watermelon snow. So those algae bloom or snow surface after the onset of melting. And so in different life stages, the snow algae can show different color from the green color to red color because of the change of their pigments. So for mapping snow algae, we had already had studies use remote sensing to actually get those red pigment because they are quite obvious from the imagery data. They have those red, very bright red, those pigmentation. But for the glacier algae, they are kind of more like a brownish, grayish color. They look like dirt. If you really use like visual color remote sensing bands, it's really hard to differentiate those glacier algae from those, other like dirt materials. And also the glacier algae, they are not grow on the snow, they kind of grow on the bare ice, so have very short growth season. So they generate what we call dark purple gallon pigment, and they use kind of as a sunscreen to protect them from the excessive radiation from the superglacial environment. So we can say, I always say they like to get tanned. So based on those field knowledge we get about glacier algae, so if there are possibility, we can map them from space, from satellite data. So one critical question to use satellite data to mapping, we have to know the biomarkers and also the spectral signatures. So this is kind of based on field study. So they kind of get the samples of glacier algae and kind of extract out the pigments of different pigments. And those glacier algae, their pigments is mainly dominated by this brownish pigment. And this kind of shows their absorption of this pigment in of radiation. So they have a very spectral signature in this ultraviolet region, and then they absorb some of the blue and green light. So that's why they look like a brown color, but they have do not absorb too much in red and near infrared. But the issue is that if we really use this as a biomarker to detect glacier algae, their spectral signatures in this ultraviolet region really not in the spectral rate of most satellites. So talking about the secondary, their pigment chlorophyll A, and chlorophyll A has widely used for study the algae blooms in aquatic systems in lakes, like we study those green algae. So similarity between those green algae in the water and also the glacier algae on the ice surface, they both generate chlorophyll A for photosynthesis. So we can actually, instead of use those brown pigments, we can actually still use their chlorophyll A pigment spectral signatures to actually extract the glacier algae. So this shows like different pigments, their absorption spectrum in this from purple blue to red light. So chlorophyll A, basically they absorb most of light and blue bands and also absorb the red light. So they have in visible light, they're gonna show like they don't absorb too much in green, so they're shown as a green color. But in near infrared, we also have observed very dramatic red edge, this kind of signature. So the problem is that if we use chlorophyll A as a biosignature or to detect glacial algae, this glacial algae really shows the chlorophyll A spectral signature. So this is kind of based on the field spectral measurements of glacial algae in compared to other materials, snow and clean bare ice and some water streams. So we do found they actually, if you look at it very closely, they actually have spectral signatures of chlorophyll A in the red wavelengths, also near infrared wavelengths. They have, even though this this edge cannot compare to really to our vegetation forest, those signatures, but this shows these signatures of chlorophyll A. So because this, these characteristics, then we can actually go back to the question, if we use satellite data to study this phenomena, can we actually use long-established legacy, ocean color satellites to do this job? Because we have long-term observations of ocean color satellite to study the chlorophyll A concentrations in the ocean. Can we actually also use the data for study the algae or the ice surface? So if we look at the ocean color satellites, we have to focus on several characteristics. First, if we detect very small volume chlorophyll A shown by glacial algae, we have to have very narrow wavelengths, So the ocean color satellites, this shows spectral response functions of Sentinel-3 OLCI sensor and also MARIS or board or the MASat. So those two sensors are designed mostly for ocean studies. So if you look at their spectral response functions, they have very nice spectral resolution in this chlorophyll A absorption and reflection this range. Compared to that, if we use LED observation satellites, they have generally have a higher spatial resolution, but they have to sacrifice certain spectral resolution. So the wavelengths are quite a broad band. So it's actually this narrow band wavelengths of the ocean color satellites actually provide very nice opportunity to capture this glacial algae spectral signature in this region. And also because ocean color satellites, even though they have very coarse spatial resolution, but they have very frequent the temporal resolution. So there's potential we can actually mapping the glacial algae blooms over one season with very high temporal resolution. So the first sensor actually we looked at is a Sentinel-3 OLCI. This was a comparatively new sensor that was launched by ESA in 2016, and it carries this OLCI had very nice bands in red and also near infrared region. So if we get to those sites that was found to have glacial algae blooms in the summer by fieldwork, we can actually see very nice correspondence of this Sentinel-3 spectrum. In this, we can see this have very nice, this reflection peak in around 710 nanometers, and also absorption in like 670 nanometers. So we like this. We can actually use as these reflection features to quantify how abundant, what's abundance of this glacial algae. And another sensor, after we actually studied the use of Sentinel-3 sensor, we kind of curious, can we actually tracing back, get a long time series of data? So we look at the Sentinel-3 is built actually upon the design of MASat MARIS data. So this MARIS sensor has a nice coverage from 2002 to 2012, and we also looked at this data, and this data also shows quite a nice spectral signatures of glacial algae. So those green lines, they are all field spectral. Actually, scientists collected in situ of those algae covered areas, so you can observe this spectral signature very obviously. So that means actually satellite data does catch this spectral response of glacial algae, and this peak we can think about is mainly dominated by chlorophyll A pigments. Because of that spectral signature, actually, we can also use ocean color kind of approaches to actually quantify this glacier algae. So there have a lot of studies, actually well-established studies, to relate spectral indices to this chlorophyll A concentration in ocean, and we generally say the proxies for chlorophyll A concentration. So we tested actually different kind of those indices to see if they also can be useful or valid for the glacial algae abundance. Well, I cannot complain too much about the lacking of field data, but we do have very limited field data, actually, especially matching with satellite observations. But the thing is that we have those historical, like some field data we can think about, they are kind of averaged through season, kind of match their spatial variation. So we kind of put together with this this 2BDA index with those algae abundance, trying just to see if they have a fit relationship. The thing is that we actually got quite a good relationship here, but again, because we have very limited field data, we really have need to have more in situ data to actually constrain this relationship. But generally, they kind of match. No matter what, the satellite data does show the can reflect the abundance of glacial algae that match with field data. To further actually test this 2BDA works or not, we actually also compared with the spectral obtained in the field. So that in the field data, they collected the field algae abundance and also corresponding field spectral data using a spectral radiometer. So we use a field spectral data, we also calculate the corresponding 2BDA index and plot against their coincident cell abundance. Interesting, we also actually found the field data actually also support this 2BDA index. It's actually useful for quantify the algae abundance. So one actually very critical question is that because we cannot say, those dark materials are all algae. Actually, in that area, we have a lot of other materials, like we have dust and we have actually change of those snow grain size. And we also have a spectral mixing problem because the satellite, the ocean color satellites, we are using basically like 300-meter resolution, right? So the in situ data only get very like several meter resolution. How we can, solve this spectral mixing problem. So one critical question is this 2BDA index is still valid if we have other materials. For example, we have changing grain size, changing snow grain size, we have different dust, we have a kind of different dust concentration, and let's also do some spectral mixing to see if this 2BDA index still works. So to do this kind of experiments, we kind of use radiative transfer modeling called SNEAKER. So the SNEAKER can basically, you can input different illumination conditions of solar radiation and also specify the different parameters of snow grain size and also the ice parameters and also what we call, different type of dust and also different dust concentration to actually can model its albedo in different wavelengths. So using this information, we can actually also calculate the corresponding indices, like we defined like 2BDA index and other indices. The interesting thing is we found that this 2BDA index, first, they are not sensitive to snow grain size. That means, variation or the spatial distribution pattern 2BDA index does not actually affect by snow grain size. And then we also tested different dust materials, and we found that so this kind of scatter plot is actually the algae, what we say the algae, those measurements we get from satellite data. We plot the 2BDA index compares to impurity index, trying to see how it is distributed, then compare against the radiative transfer modeling. So basically the information here is that if the dust have impact of 2BDA index, it generally also have very high impurity index. That is not what we observed using satellite data. So for the satellite data, if you have this kind of range of impurity index, but you also have very high 2BD index. That means it's not caused by the dust concentration or different types. And we also conducted a bunch of experiments, if we mix those different algae percentage within area to see if the 2BD index still works. So we found that actually 2BD index also works for this different combination of this algae percentage. That means even though your spectral is mixed with between ice and algae, they still can get the algae signal from this using this kind of index. So with this kind of indirect actually validation use those past field data and also radiative transfer modeling, we can basically say this 2BD index kind of can provide a proxy of how abundance of glacier algae on the ice surface. So because of that, we can actually combine all those Maris images and all the Sentinel-3 images. We can get a very nice algae distribution map in different years. So those are kind of aggregated by year, like through the whole, the dark ice the season that we aggregated the 2BD index. So they kind of shows very nice interannual variability. So you can see in 2008, 2009, 2010, and 16 and 19, we have very high algae concentration on surface. So the problem is that what factors, can control this algae interannual variability. So we plot out against the regional climate models. We have those kind of different, short wave radiation, long wave radiation, and also snowfall, rainfall, and also temperature, meltwater content, trying to say, in high algae year, what is factors is dominating. So we found that if you have more short wave radiation, basically means less cloud, and you have warmer air temperature and less precipitation, and you're gonna have more glacier algae. This is actually very intuitive. Basically, glacier algae, they have to do the photosynthesis. So you can imagine if you have less cloud, have more sunshine conditions, they just gonna have more productivity and they have more concentration on the surface. After we kind of ensure, this 2BD index works and is valid for detecting glacier algae, and also after we, mapped their interannual variability across the space, we can also see how the seasonal growth pattern of this glacier algae. Use the field data, we basically can, for specific field sites, we can see the, the growth cycle of the glacier algae, but how that actually spatially distributed, was if there any spatial variability of this seasonal growth of glacier algae. So first problem is that can we actually tracking their growth pattern, use 2BD index versus their field data. So the 2BD index is this black line we get from satellite data. So you can see the general trend quite match with the field data. So with this information, we actually can map out, how the algae grow over the season in different kind of spaces. So this kind of shows two maps. So first map shows coefficient, means the darker color means the algae grows more rapidly, and this lighter color means less algae or grow less rapidly. So those very pinky areas, they're all very algae abundant areas. So this map shows corresponding R-square estimate. So the information we get here is actually we can quantify how much time that algae can double their population. So those coefficients, if you combine those coefficients and a number of equations, we can do very nice estimates here. So if here for this site, we have coefficients 0.0009, so that corresponding to population doubling time is around 8.85 days. And this is also have very rapid growing rate, and this is very low. So it's interesting that we actually, to be honest, this satellite data is very core spatial resolution, and we actually expect a lot of uncertainty here. But if we look into the literature, we found that the field data actually also have some observations of this glacier algae dumpling time is estimated around 7 days. So the 7 days compared to the satellite data we map about 8 days, is actually very, very close to each other. That means, the satellite data in this kind of spatial scale can actually did a pretty good job of matching field observations. So another thing is that we really care is about the surface albedo, because we care about glacial algae because we think it's darkening the surface, and darkening surface is going to cause a surface albedo decrease. So if we want to do a very nice job in terms of estimating the meltwater in this area, we have to parameterize correctly of the surface albedo. Unfortunately, the current regional climate models and climate models, we do not consider those biological impact of glacier of this surface albedo. Basically, we don't have bioalbedo scheme very well incorporated. So it is necessary to first quantify how the glacier algae change the surface albedo at a spatial scale, like satellite measurements and also regional climate models. So we also combine, the algae information we get from 2BD index, and we combine with actual, the albedo we get from MODIS satellites, and we estimated how algae population doubling can cause the reduction of algae, the surface albedo. So the information basically here is that for this site 1 means that, for each algae population doubling, we actually can reduce the surface albedo by 3.06% of the albedo. So this actually, again, somehow match very well with their actual field measurements of albedo and also the algae concentration measurements. So the problem is the next step is really, how to actually incorporate the satellite data and also the radiative transfer modeling data and also the in situ data to kind of build a very robust bioalbedo scheme for, reducing the bias in this area to model the melt and also albedo change. So here is some concluding remarks, and basically to here want to emphasize is ocean color satellites can map the glacier algae by utilizing the chlorophyll A spectral characteristics, and we can actually get very nice growing pattern of glacier algae, retrieved from satellite data, actually matching with the field data. So this is really interesting, like in the future, how we can actually reduce the albedo bias parameterizations by considering the algae pattern in Greenland. Yeah, here's some. I just put more details in those paper and also acknowledgement to the collaborators. That's yeah.
Thank you. Great talk. Very interesting. So again, right, please go to the chat, and as people are, getting their questions in, I sort of have maybe a comment and a question together. So what I want to make sure I understood it. I took one of the things I took away, sort of especially with the application towards planetary science, was that that a high spectral resolution instrument was really key to finding these unique features. Is that correct?
Yes, yes, it's correct. Especially if the biosignature of chlorophyll A, even though we already know there's glacier algae, there are a lot of glacier algae, but when you look at the space, they are actually very tiny spectral signatures. So you have to have very narrow spectral bands. So I really like flag mission. I'm just curious, is there any possibility you also carry a hyperspectral imager, because that can really I don't know, correlate your your sampling those plumes and also the remote sensing observations. I feel
yes, missions definitely do that. I would actually ask Shannon to pipe in about whether or not what level of spectrometer was sort of maybe planned for Orbilander. But just one other question before we do that. So do you know, does anyone sort of Has anyone tried to do what we would do as planetary scientists? Have you looked at that data from it, with the narrow bands for an unexpected signature, sort of try to pull out something, that could also be related to microbes, that wasn't expected? Sorry, did you hear my question?
Oh, that's to me or
That's for,
Shannon?
that's for you. Because you focused in on chlorophyll A. Have you ever sort of said, there's an interesting band here that I wasn't expecting, or do you know if people are doing that at all?
You mean chlorophyll A, or
Not something that you might not expect, but looks, biology or organic or something where discovery style science, as though you were planetary scientists.
I don't know. I don't know actually.
That's okay. I'm
Yeah.
taking up too much time. Shannon, please answer her question.
Yeah, certainly you could envision sending a fun spectrometer to characterize the surface. We did not end up putting an IR spectrometer, but focused more on like the thermal wavelengths, because we wanted something to help us characterize the regolith properties, again, to build up our confidence that we weren't going to send the landed portion of the mission to something too fluffy or too warm or too soft or too hard. So we didn't focus on like surface composition characterization, because we figured we were doing that with other metrics, or other measurements of the plume.
So what's the
Thanks.
spatial resolution you can get for thermal imager?
To be honest, I don't remember what we ended up with off the top of my head, but it depends on a lot of different factors, like what your altitude is, which kind of spectrometer you're using. Yeah, I would have to go back and look. I'm sorry.
Okay, quickly try to take a question or two from the chat, although we're just about at time. I guess here, so a comment. One thing is that algae is vertically distributed, and all your measurements are cells per milliliter. For snow algae, we've started using cells or pigments per meter squared to make the measurements coherent across different depths. So you have a comment sort of about that depth issue?
Yeah, depth issue, definitely no way. The satellite is just so tall. There's no way you can get depth measurements, only the surface approximation. And about the fluorescence, that
wasn't my question. Sorry, can you hear me? That wasn't my question. It's that for the ocean, we have chlorophyll per meter squared, right? So we integrate all the chlorophyll and then we relate that to the satellite, which is something you should work towards in ice too, because the measurements people make depend on whether they excavate 5 centimeters or 10 centimeters. So you should do like excavate all of the algae and then do a pigment or chlorophyll per meter squared to make these measurements. I've looked at them in the literature and you can't cross studies because somebody measures algae in one depth versus another depth. So my point is it doesn't necessarily see all of the vertically resolved pigment, but certainly you can relate the surface measurement best if you know the whole vertically excavated amount of pigment in that surface area. So that's how we do it in the ocean too. We have chlorophyll per meter squared is kind of where we go for our productivity models. Because the surface.
Thanks for the clarification.
Yeah. But I wanted to know if they fluoresce because it didn't. You have that little bump at 709, but did you see any chlorophyll fluorescence at like 680 due to chlorophyll in your measurements?
So maybe I put out this third paper here. I listed. You can see this third paper is actually. This is a paper maybe actually can answer that question, that actually kind of want to use this solar fluorescence, relate to what we see from ocean satellite. We do see some correspondence.
Okay, and we're over time.
Thank
so
you.
I want to just unfortunately please reach out to Shujie if your questions didn't get answered. And thanks again to both our speakers for really great talks. Just take one more minute and say that this is the final speaker series for Michelle, Audrey, and myself as hosts. It has been a pleasure putting these together for the NOW Research Coordination Network. And thank you all for your attendance over the past 2 years.